Okay, hello everyone. Welcome today for, I forgot the important part, to also say bonjour tout le monde. Bienvenue à cette occasion spéciale donc, qui est une conférence associée au prix CRM-Fields-PIMS. So this is a CRM-Fields-PIMS distinguished lecture. The CRM-Fields-PIMS prize is the most important prize for research achievement in Canada in mathematical sciences. It was started first by CRM and the Fields Institute in the middle of the 80s, and it was joined by PIMS at the beginning of the years 2000. And so today we have a lecture by the recipient of the 2025 prize, who's Leah Edelstein-Keshet. And she completed her PhD at the Weizmann Institute of Science in 1982. And she joined the UBC faculty in 1989. And she's currently emeritus professor there. Dr. Keshet has received numerous prizes for her work, including the Krieger-Nelson Prize of the Canadian Math Society and the John von Neumann Prize of the Society for Industrial and Applied Mathematics. And she became a Fellow of the Royal Society of Canada in 2023. Dr. Keshet is considered a pioneer in mathematical biology, and she made contributions to understanding of biological phenomena such as cell polarization, cell motility, and collective biological motion. This is all very strange to me because I'm a topologist, and I'm very impressed that about all of these things, and I hope to learn much more about them today. So her highly influential research integrates mathematical modeling, computational analysis, and experimental collaboration. And she will speak to us today about mathematical biology of cells, structure, movement, and collective behavior. Thank you very much.
Wonderful. So first, thank you very much for having me here, also for CRM, for PIMS, and Fields for this great honor. It's very humbling, in particular when one looks at the name of previous recipients. So it was really a surprise and stunning. Thank you so much. So today I'm going to tell you a little bit about some of the things that I've enjoyed working on over the years. But just before I do that, I want to say a few words about what is mathematical biology, and also how I got into it and what was my journey to get into this field. So you can think of it either as an area of math application or application that fosters new questions in mathematics, new ideas, new topics, new problems for us to work on as mathematicians. You can also think of it as a branch of mathematics that can be used as a tool to help biologists and to help them solve their mysteries. So a few words about how I got into math biology, and you'll see it's quite a natural fit. So this is a picture of my mother working in the lab in Halifax at the National Research Council in the 1970s. And this is a picture of my father working. He's actually proving theorems in his head, and of course later he wrote them on pieces of paper. But obviously mathematics has this beautiful attraction. You don't need a lab. You really don't need much other than paper and pencil, and away you go. Okay, so a little bit about my trajectory, some of the problems that I worked on, and you'll see that there are interconnections between things I did in the beginning and things that I'll describe later. So I was a graduate student in Israel with Lee Siegel, an applied mathematician. And from him, I learned about self-organization. And in particular, this is one example of a kind of social amoeba that can either live on their own or aggregate, stream together, as shown here, and then eventually form all these really beautiful, complicated, three-dimensional, multicellular structures. And one of the questions was, how do these things aggregate and how do they get together? So from Lee Siegel, I learned about the PDE model that was so-called the Keller-Siegel model. It is a set of PDEs for the density of the cell, the density of a chemical attractant. The cells undergo random motion and they're attracted to the chemical by chemoattraction. And so it's a pair of PDEs. One of them you'll recognize as having a diffusion term and a transport term, so-called chemotaxis, whereas the chemical is just reacting and diffusing and being secreted by the cells. This, by the way, this model, which here I show in one dimension, has been intensively studied after a gap of about 10 years when it was proposed. It's been studied in 1 and 2 dimensions and higher. In 2D, this model can lead to blow up. And these are fascinating mathematical problems. You'll see later that one of the reasons why this led to blow up is the fact that there is attraction, but there's nothing keeping the cells apart. They can pile up together. From Lee Siegel, I also learned about theories for pattern formation, namely theories that date back to the 1950s, when Alan Turing proposed the idea that by reacting and diffusing, a pair of chemicals can give rise to spatial patterns. And this is a typical set of PDEs that is studied in that general realm. And in particular, if one of the chemicals is a local activator, it has a small rate of diffusion, and the other is an inhibitor that has a faster rate of diffusion and acts globally, then you can get patterns to form. And those patterns have been invoked to explain many beautiful things biologically. So both pretty patterns on fish or birds or butterflies or fur patterns, as well as simple polar patterns like this billy goat. And I'll return to this idea a little bit later in the context of a cell. All right, so now let's flash forward after my PhD. Some people in this room, at least one person that's here, Leon, remembers these amazing conferences. Gordon conferences where all people who were interested in math and biology got together. It was a pretty small field at that time and talked about problems we were interested in. And there I met a genius, Bart Ehrmantraut, who also became a very good friend of mine. And we started working on a bunch of different problems. And one of the problems that I wanted him to be interested in and we got into is a problem related to multicellular behavior. So here what you see is not very good images of individual cells that when their density gets very high, they somehow orient themselves in parallel. They form these nice parallel arrays of cells. So this is roughly hundreds of cells we're seeing here. And we asked, what is it that allows this system to self-organize in these sort of ways? So we hooked together a model which had both a PDE integral component and agent-based. I'll show that later. But basically that model described what cells are doing in order to understand this process. So let's look a little bit at the underlying assumptions. We assumed that every cell has some orientation, it has a polarity, it undergoes some kind of random motion and reorientation. We're specifically interested in the angle and not so much in the spatial position at this point. When cells come into contact, one of them may align with the other. And they form these clusters, or they bind one another, and they can leave and arrive and bind to such clusters. So that's really what these equations are describing. Now, there's a feature in this equation which encodes the angle dependence of the binding and alignment. So these stars are actually convolutions. And what they're saying is that, for example, two cells may align each other, align and bind if they have the correct orientation. So in more detail, these terms that are in the equation are actually convolutions of cell density with the neighboring or the surrounding cells. Space is not being considered. We're thinking of everything as basically in the same spatial domain. And so each of these terms is one of these non-local type of models. And K here, the kernel encodes a probability that cells that subtend a certain angle will turn and stick to each other and form a little aligned grouping. So here's an example of a kernel of that sort. What this says basically is that cells that are almost aligned or at some acute angle, up to about 45 degrees, have a high probability of alignment, but those that are almost orthogonal to each other, they basically just don't pay attention. They just continue moving. So again, angle dependence. So the analysis, this is, I should point out that this is a system of integral PDE, so it contains some partial derivatives, but it also contains these non-local terms by virtue of the convolution. So at that time, that was in a way a little bit new. Not that many people looked at these sort of non-local type of models. The analysis that we looked into was if the cells are initially uniformly distributed in angles, is there some kind of instability that would tend to promote this self-organization into this angular pattern formation? So as you may imagine, this is really a problem to which we can apply so-called linear stability analysis. So I'm sure many of you are familiar with this. We have a uniform steady state, and we look for small perturbations of that steady state. And those perturbations have some wave number K, and they have a growth rate lambda. So if we can find conditions, or we can find wave numbers such that lambda, the growth rate, has a positive real part, then we have stability, and we have some kind of growth of a pattern in angle. So a lot of algebra later, you arrive at a condition when this instability happens. And that condition is in terms of the Fourier transform of the underlying kernel, alignment kernel. And also it contains a term that comes from the random reorientation with a coefficient that depends on all of the parameters that appear in the problem. Those parameters include rate of binding, random reorientation, and they also include the total number of cells, or the total mass. So you can analyze this in particular for the kernel that I mentioned earlier, where cells tend to align at acute angles. You can analyze this inequality by looking at those two things at the two sides of the inequality. The Fourier transform gives rise to a kind of bumpy looking set of peaks, and the term, so this whole thing is the bumpy set of peaks. This thing over here on the right is a parabola shown dashed. And you can pretty well see that as this quantity A decreases, the parabola starts to be broader, which means this inequality is more likely to occur. So in particular, if the mass or the number of cells gets larger and larger, it's more likely that instability will take place. So that gives you a little bit of a flavor of the kind of argument that was made here. And of course, simulations and agent-based models dating back to the 90s. So you can see how much we've improved in visualization. Yes.
So in this model, just to make sure I'm understanding, instability meant that the cells did align themselves?
Yes, that you began to see patterns in angle, in fact, as shown here, right? So you would start off with something that was nearly uniform in angle. And when that inequality was satisfied, when you had enough cells around, you would tend to begin to grow these peaks. A lot of cells were pointing this way or pointing directly 180 degrees to it. And you see the same thing in these very primitive printouts of what agent-based models looked like back then. Okay, so back to Canada and UBC, beautiful campus. I was very lucky to get a job there in 1989. And at that time, I started to get interested in a problem that had to do with moving cells. So this is a classic picture or movie of a white blood cell that's navigating in between red blood cells, and it's actually chasing a little bacteria. And so these are cells in our immune system that have to be very, very good at detecting infections and following and killing pathogens. So I will point out that when you look at the movie, the natural question is, how can this cell do what it does? It's got a front that can sort of detect where to go. There's a back of the cell. It's always deforming. It can turn and so forth. How does it do it? But back in the early 1990s, this was still very much a project for the future, because at that time, people were looking at components of the cell. And in particular, they knew that there's a certain component, the so-called actin cytoskeleton, that is very important for being able to push the front of the cell forward to allow it to move. So what is actin? Actin can exist both in subunits and also as long filaments that assemble those subunits into polymers. And back in the 90s, people were highly interested in different structures that actin could form, either bundles where the actin filaments were more or less in parallel, or these sort of orthogonal and basket weave type of meshworks. These are from the Borsey lab, and people were very interested in understanding these structures. So at that time, this, by the way, is a photograph I found when I had to clean out my office. So I retired recently, buried under a pile of stuff, and it shows, believe it or not, me and my first PhD student. Yes, I looked like that back then. And also Alex McGillner, who was part of this whole story. So we asked the question, what causes actin filaments to align in these big strands or create these meshworks? And you, surprise, surprise, much the same kind of methods proved to be useful. So I won't go into details here, but again, you find that there are convolutions. There are things that describe the tendency of filaments to bind to one another. And depending on additional proteins that facilitated orthogonal or bundling binding, you would get one or the other kind of crystals. So that's just to show that, you know, once you get familiar with a hammer, a lot of things look like nails, right? So it was in a way an extension, but taking it from the realm of cells down to intracellular structures. So on to a little bit more recent work on, again, the cell polarity and motility. One question that arises and was kind of talked about a lot in the early 2000s is, how do cells manage to figure out that they should have a front over here and a back over here? So I'm not talking about bacterial cells that are rigid, I'm talking about neutrophils and white blood cells that are always deforming, and they need to simply know which way to go. They have to polarize, they have to have a front and a back. So cell polarity was a big question that biologists posed, and many people got into this general idea of how can we explain it. So what I want to tell you very briefly, because there are a lot of biological technical details, is that in order for a cell to have a front and a back, there's actually an internal reaction-diffusion system that forms a pre-pattern. And that pre-pattern tells actin where to go, and the actin tells the cell where to move, and so on and so forth. So this thing that we learned about the Turing pattern-forming system started to become important. Now, in real life, it's very complicated, right? Actual cells have all this stuff in them, and this is really a simplification of what cells have in them. And somehow we have to understand how this works. For our purposes, we think of this as a little cartoon of cell detects incoming signals, funnels the information through proteins into a kind of pattern-forming subsystem, and that subsystem tells, there's a readout, it tells the actin filaments where to go. And we decided to simplify it in order to understand something. So we looked at one of the many, many proteins in this pattern-forming module, but we did use some of the essential biological aspects to help us explain what was going on. So essentially, so now looking at a slightly larger picture, essentially, there are two forms of this protein, which is going to signal where the front will be. And it turns out that you can think of this as a slow, fast system. Now, this little icon will reappear several times. So I'm showing it large here. And here it is again. So back around 2008, with PhD student Sasha Jilkin and a very talented postdoc, Yuichiro Mori, we looked at PDEs that describe this sort of protein that has two forms with very vastly different rates of diffusion. And that's because one of them is embedded in the cell membrane, which is highly viscous. The other is inside the cell, and therefore it can diffuse very rapidly. And so it turned out that this set of PDEs with some minor assumptions, which I highlight here, gave us an explanation of cell polarity. So first, what are the important features of these reaction-diffusion systems? First is that the total amount is conserved. So in other words, the activation of one of the proteins is at the expense of the other one that acts like a kind of global, fast diffusing messenger. So that's one thing. I already mentioned that the different rates of diffusion is important, and some kind of nonlinearity and positive feedback in the lingo of biologists is also vital to get the system to work. And when it works, here's the way it behaves. So it forms a wave, it invades into the domain, and it freezes in the middle of that domain somewhere. So even though the simulation will keep running, this wave freezes. In other words, it distinguishes two ends. One end would be a high active zone, the other end would be low. In other words, it can create a polar pattern. And that's precisely what we need in order to explain something like cell polarity. Okay, so now just to put together what I told you, how does all this fit? So I should mention that just before doing so, that this thing has been nicknamed, this model, wave pinning, because of the fact that there's a wave that freezes, that pins in the middle of the domain. So the idea is that wave pinning or something like it will cause RAC, or this protein, to form a pattern. It in turn signals to the actin assemble here, and the actin then causes the protrusion of the front edge of the cell and the motion of the cell. And this just shows that, in fact, yes, you can simulate this basic idea, and it will, in fact, give rise to both pattern inside of this deforming domain and motion. Let me just check how I'm doing with time here. Okay, so just before I move on, this and variants thereby that include more detail or less detail, we spent many years looking at these. Many postdocs, many graduate students had the fun of coding up these simulations. And let me mention that there's a bit of a challenge here, because what you're doing is you're solving a set of PDEs in a domain that's constantly deforming with norm and boundary conditions. So we know that that can be difficult. That can be something that's challenging. Okay, now just recently, with both the advance of software and with a very talented master student, we're able to simulate cell motion in a simple way on an open source platform called Morpheus. And so my student Jupiter Algorta, just for fun, coded up this kind of a moving cell, and he even has, if I'm not mistaken, a little bacterium that the cell is chasing and moving through obstacles. And based on the fact that he could get this running in this software, we were able to attract collaborators who are actually studying real cells. So Orion Weiner and his former postdoc Jason Town. Orion is I'd say one of the famous cell biologists working on cell motility, and in particular on white blood cells. And the beauty of biology is that it's a field that makes progress in leaps and bounds. So nowadays, what Orion can do is he can take a white blood cell that's been slightly transformed into his little special cell, and he can guide it by using light. It's called optogenetics. So you have a laser that basically guides the cell to either move left, to turn, to move backwards, and so on. And they have a fantastic set of data, and we started working together on this topic. The first model we looked at was precisely the same, and then we looked at small variants. So it's the model that can explain polarity with a couple of other little features added to account for some of the observations that they had. And amazingly enough, the data set is so good that we can actually fit parameters of the model to the real cells. This is, I think, the first time that, to my knowledge, that wave pinning and these other variants will fit to actual cells. And we can also account for all kinds of strange behaviors. This is the real cells, over here it's simulated cells. So there are certain experimental protocols where the cell is stimulated locally to turn in one direction and then stimulated globally, the whole cell is illuminated, which causes the cell, the experiments, to turn the other way. And we were able to account for that with slight variants of the model that I described to you. So I know for mathematicians, it doesn't seem like a big deal, but for biologists and for math biologists, it's like, yeah, they were. So no topology, no big, wonderful function spaces, but for us, it was very exciting. I want to mention that not just simulations, but also we do try and do some analysis and try and understand the behaviors of these systems. So typically this might be a set of 3 PDEs. And now we're going to look at them on the edge of the cell, a one-dimensional periodic domain. One of my students, Jack Hughes, is just finishing a PhD in which basically he did a full PDE bifurcation analysis of the PDEs that describe this model. So this model is very much based on what I've already told you, with one small feature, and that is that the actin also inhibits or works to inactivate this special protein called reaction. And it turns out that this system is quite interesting because not just polar solutions. So here, these are space-time plots. Biologists call these chimegraphs, but not only spatial polarity, but also traveling waves exist in this pair of PDEs. And Jack was able to show that they can coexist. There are some parameter regimes where a cell could do either one of these behaviors, could either exist as a polar cell, or if you perturb it a little bit, it could go into these waves. And in fact, that is also seen biologically. Working together with Jupiter, they were also able to show that this implies different behaviors, polarity, cells that turn or have various kind of waves moving around their perimeter. Okay, so much. I've told you a little bit about early non-local models. I've told you a little bit about cell polarity, cell motility, and our efforts to understand them. And now I want to build on that and describe collective behavior of many cells or many agents and so forth. So I originally had many slides here where I showed how we applied these to ducks and to all kinds of different examples. But in view of time limitations, I'm going to just show one example before I go into the last piece of the talk, which is slightly more mathematical in flavor. So, for example, we did some work on collective cell invasion, for example, in cancer, based on a paper by Andrei Levchenko. And so a master student of mine took something that was intracellular. Again, you see the protein RAC makes an appearance here, where this circuit can either govern the tendency of cells to move fast, or on the other hand, the tendency of cells to stick to one another. And she showed that you can account for breakage of collective many cells under certain conditions, breakage away from a group of cells. So think of cancer metastasis, etc. Okay. But I really want to focus on one final example before I end, and that is on recent works in the last 5 years or so, looking at cell clusters. And this last project was actually motivated by biologist Paul Kulisa, who also was an aerospace engineer originally and thought that biology just so much more interesting than rockets. So he works on embryogenesis, and in particular told us about a cluster of cells where the cells don't actually stick to each other, but they like to make a nice little loose swarm, and together they will go migrate and do various things. The question is, how do they do it? And his idea was that it's likely that there's some kind of signaling between the cells involving attractants and repellents, etc. After we visited his lab, I found a really nice paper from this group of Jose Carrillo. I'm sure some of you have heard of him. He works on PDEs and all kinds of very interesting thing. And this is Ruth Baker. And basically what they looked at is a non-local model for cell density, where the cells, here rho is the density of cells, let's say, and the cells are moving at velocity v. That velocity is essentially coming from the gradient of some kind of potential that the cells are creating in some way. And in their paper, what they show, so again, this potential, it depends on sort of a non-local neighborhood of cells. There's a convolution here, just like things I talked about earlier, with one important difference, which is that now it's a potential that causes a velocity. Its gradient is a force, it causes a velocity, whereas in the original examples I gave early on in my talk, those convolutions were more like instantaneous transitions from one type of cell to another. In their paper, what was also nice is that they show that this non-local model, which has an integral in it, can also be approximated in some instances by a PDE of higher order, by basically a fourth order PDE, where these coefficients depend on moments of the kernel. So we took what they did and we made the following connections. So first we looked at a specific type of potential or kernel, exponentials, so-called Morse potentials. And then we showed that actually that comes out very naturally from a generalization of the Keller-Siegel model that I told you about early in my talk. And we classified the steady state behavior and showed when can you get a cluster. So just very briefly on the tip of the fork, I want to show you how that goes. So this is the generalization of the Keller-Siegel model, where you've got cells, density rho. You have two types of chemicals, not one. One is an attractant and one is a repellent. And the cells secrete each one of them. And the cells are attracted to one of them and repelled from the other. So with that in mind, and using the fact that you can separate time scale, chemicals diffuse really quickly relative to the cell motion, you can basically solve this steady state chemical PDEs using the Green's function method. And lo and behold, you get, in one dimension, you get exponentials. A picture of what that looks like. So think of this as a single cell with one chemical field around it that it is secreting. So instead of this non-local model, instead of the original model with 3 PDEs, you can boil it down to a single non-local model where the potential depends on these chemicals that the cell secretes. Not just that, but you can identify parameters in this system, diffusion rates, secretion rates, and so on, that make up the ranges and the magnitudes of each of these components, their repulsion and the attraction. So just visually, if you have a cell at the origin, it will create kind of repellent and attractant profile around it. And the net potential that's difference of the two will look something like this, most potential. And then you can completely understand and analyze the system by using the same kind of local approximation that was suggested in the Carrillo paper. Now, these coefficients turn out to be directly related to parameters of the problem. And so now you can actually get an explicit solution for the shape of the cluster. And that solution depends on the Morse kernel. If we rescale, if we look at parameters that are the ratio of the repulsion to the attraction magnitude and spatial range, then we can now characterize exactly what's going to happen to that cluster. So only in certain parts of this parameter plane are you going to get a viable cluster. In other parts, you'll get collapse, net repulsion, or possibly fragmentation. And these are things that you can verify by simulations. Here again, showing space-time, space on the x-axis, and the y-axis, showing that in one region of this parameter plane, you get a cluster that forms and persists. In other regions, you do not. The cells either spread apart or potentially not shown here. There are regions where the cells collapse according to the continuum. And furthermore, you can, with doing all this analysis, you can go back and say, what does this imply about the actual cells? So you can rephrase your conclusions in terms of parameters that are actually applicable biologically, such as ratios of rates of diffusion of the repellent and attractant, or of their rates of decay, or of the rates of secretion and chemotaxis of the two cells. So I think I'll skip this in the interest of time, and I'll mention that you can, of course, simulate this with agent-based models as well, and show that there are regimes where everything collapses. There are regimes where you get sort of nice distribution of cells, fragmented and collapsing cells, and so forth. Okay, so I'm basically done. I just want to summarize. There were a couple of themes that came up that I think are worth just noting one more time. That in continuum models, we've seen that both local PDE as well as non-local integral PDEs have become very useful, and that sometimes there's a way of exchanging one for the other in order to get some analytical insight. I haven't talked too much about discrete models. I showed one or two old and newer simulations, but we've certainly done our share of these type of models as well. Another theme that I wanted to quickly touch on again is this idea of self-organization. And you've seen that in two separate contexts, one being the so-called Turing reaction-diffusion PDE system, where the more classical version is to have a local activating chemical with a global inhibitor. But our wave pinning was actually local activation and depletion of a pool of stuff that you need in order to create a polar pattern. So that was one class. But then there's this other class of self-organization by transport or chemotaxis of cells. And there really we found that in order for cells to form a nice cluster, you need to have them locally repel each other so they don't all cluster in a very, very tight group, but also to have some kind of longer range attraction. So finally, I hope I persuaded you that biology gives us a huge variety of rich phenomena that are just waiting to be addressed mathematically. And I've certainly had a lot of fun doing this over the years. And I'm really grateful both to PIMS CRM and Fields, to my great colleagues at UBC to answer for funding, and also to a great many young scientists who've been very helpful all along. Thank you very much.
Thank you very much for this wonderful talk. So maybe there are questions. Yes.
First of all, let me thank you very much for this beautiful lecture. I very much enjoyed at the beginning the way you described how you became a mathematical biologist. Your mother was a biologist, your father was a mathematician, and you integrated the two and became a mathematical biologist. Now, I also played with the idea to become not to become a mathematical biologist, but to do something in this direction. And I was at Cornell at that time. And speaking with John Guggenheimer, he told me, but Dana, there are only 25 people in the whole of the continent who do this. So this was sort of a discouraging remark. Then this was happening in the 1980s. And now I see that you were one of the 25 people and you stick with it. Bravo. Finally, I want to, because I always thought that the flow, the current of ideas come from mathematics into biology. But you have a phrase in your abstract which says the following. It can motivate new mathematical problems. So I wonder if you can tell us something about that you encounter these new mathematical problems in your career.
So I think, first of all, we should give credence to Alan Turing, who was motivated by morphogenesis and trying to explain how pattern forms in biology. And he came up with this idea of reaction-diffusion can foster. So that lay dormant for decades.
Morphogenesis, you talked about
Morphogenesis, for creation of form and development and formation of pattern in embryos, in animals, in birds. So that's one really important example. So there's a huge amount of PDE work on reaction diffusion systems. And certainly I've not contributed much of in the way of that whole big field. I think from my perspective, wave pinning and the fact that you could ask a natural biological question, how do cells polarize? What gives them a different front and back? How can a chemical inside a cell or protein create a distribution, high activity at one end and low at the other? And out of that comes reaction diffusion, waves that stall, all of these things that we see.
Yeah, I was wondering in relation to morphogenesis and wave scaling. So one of the big problems in biology is the problem of scaling. Like you have cells of different size, embryo of different size, and one of the big challenge, I think, for us is to understand how this is possible, because usually when you are reaction diffusion, you kind of fix a scale. So I was wondering, wave pinning, like how does that behave with respect to scaling? Like, can this be a good model for scaling, or can you change it to make it a good model for scaling as a function of the size of the cells?
Right. In wave pinning, the important the ingredients that allow for that to explain polarization are basically the ratio of the two rates of diffusion. And if that's okay, if that's adjusted, and the other conditions are met, then you'll have wave pinning. So now cells, well, they can exist everywhere from 1 millimeter in diameter, if you're looking at big frog eggs, down to 10 or 20 microns in diameter. So will wave pinning by itself work all across this? Possibly not. But in the real cell, remember that there's a lot of other fine tuning that happens. Wave pinning can work in a pretty large range But again, there's so many other things that allow you to extend that range and to fine tune and make sure that things work properly.
So are you saying that maybe that the sort of the diagram you showed us with zillions of different mechanisms, maybe there would be many, like several wave pinnings that are joining up a few different scales to each other until they reach those sort of scales changes that you would see in practice?
Right, so I showed this diagram, right? And even if we include not just one, but two, rack and row, which I'm going to have trouble, here it is, rack and row, and the fact that they're mutually antagonistic, we already get a more, and if we include a few of the other bits and pieces, it's not just stable, it's also able to integrate signal and smooth them out. And so there's a lot of little knobs. And really what we did is we just took one and looked at the very, very basic rack type protein and what it can do in its own. We're not saying that the real cells do that, but on the other hand, in these white blood cells that Professor Weiner is looking at, a very simple model that includes rack and two other things already accounts for it.
Hi, Leah. Thanks for a great talk. I was wondering, in the collective cell behavior, which have you integrated also cell plasticity? So the movement of the front within that, because we know, for example, stem cells are more plastic in certain types of cancer. And how does that potentially change some of the modeling or the results?
Yeah, not in that particular study. So that was done by a medical student during COVID under, we know what conditions those were, and so we were lucky to be able to do it, and she has moved on. So I haven't gone back to that, but really interesting question. Maybe we can discuss it sometime.
So thanks for the lovely talk. So we've seen huge evolution in mathematical biology over our careers. But for many of us, I think we still have this fascination with mathematics and fascination with biology. So when you see young people now who are at early stages in their careers that have this fascination, do you have advice for what they should learn, how they should spend their time? Because it's hard to know what to do and how to do this, especially since many universities still have departments which do not encourage always cross-disciplinary studies.
True. Yeah, very true. And luckily UBC has been a place where this was possible. Although I have to say that even there, it took some time, right? So in the 1980s, no, I was not one of those famous 25 people I were. No, not at all. It took. You mentioned working 70s. Yes, but I was not known. I mean, that was kind of like big, right? What to advise them? Well, I think advice number one is learn your math, because it's way easier to learn math in a structured way in university and pick up the biology by reading and talking and so on, than the other way around. So that's number one. Number two, listen to seminars in biology, go talk to people who are doing interesting things. Come to some workshops. I know the center, your outfit has had wonderful summer workshops, right? Where people can come from elsewhere and learn how to tackle modeling, how to think about dynamical systems, and look out for interesting problems. I think you've got to learn the concept, learn the math, but also be open to problems.
Okay, let's see if there are any other questions.
There
I
are
think
questions
not.
online.
Yes, online. Please go ahead.
Hi, Leah. Great talk, by the way. This is Anmar.
Hi.
I just have a question about the wave pinning phenomenon that you've highlighted. One of the things that I've noticed, at least in the simulations you've done and the experimental work, there is always a stimulus that can induce a change in directionality within these cells. Is it possible to actually combine wave pinning with this change in directionality in using an endogenous or intrinsic mechanism within the cell without an external stimulus?
Okay, good question. Is it possible to feed something else that's happening inside the cell into wave pinning and getting the cell to polarize? Is that what you mean?
Yes, that's correct.
Yes, I think so, although I'm not sure I can come up with an example on the spot. But certainly there are a great many arrows that point to influences that go towards these special proteins like RAC, right? So a lot of things happen because the cell receives stimuli from outside, but once the cell has reacted to that, there are a lot of active and inactive things inside the cell that can influence these things. So I think the answer is yes, but I'd have to think about it a bit more. But thanks for the question.
Thank you.
Great to hear you.
Okay, any other questions maybe online or in the room? No, thank you very much again.
Thank you.